Good evening. Today we are going to discuss about uh, laboratory diagnosis of bacterial infections. First, let us see what is specimen collection. Specimen collection is nothing but collecting the required bacterial samples which obey the following principles. So the first one is standard precautions should be followed for handling all specimens, and before antibiotics started this, the cultured specimens should be collected prior to any use of antimicrobial agents. For example, if a bacteria is already interacted with any antimicrobial agents, then such bacterial samples are not allowed. So we want bacterial samples to be collected which does not undergo any exposure to antimicrobial agents. Contamination should be avoided. Contamination, indigenous flora should be avoided, especially while collecting the urine and blood culture specimens. So the contamination from the plants should be avoided so that while collecting the bacteria from urine and blood culture specimens. Swabs are also convenient. Cotton swabs are also convenient, but they are considered inferior to tissue and body fluids. Next, in container. The container in which the specimen is to be collected must be sterile, tightly sealed and leak proof so that air won't enter inside and it won't contaminate the given bacterial sample. Next, labeling. So labeling must be there. All specimens must be labeled with name, age, gender, treating physician, diagnosis, type of specimen, desired investigation name should be mentioned on that container. Next, specimens which are contaminated or wrongly labeled must be rejected. If anaerobic culture is made, then proper anaerobic collection containers with proper media should be used. Okay, so these principles must be obeyed, must be followed while taking any bacterial sample. So the specimen is need not be sent in the container containing formalin. That is very important. And then next we will be discussing about specimen transport. So the specimens, as soon as they reach the laboratory for further processing, uh, so the specimen must reach the laboratory for further processing as soon as possible. So the required transport media should be used. So specimens that require an immediate transport, that is less than 15 minutes, thus the cerebrospinal fluid and body fluids, ocular specimens, tissue specimens and bone specimen. For these specimens, there should be immediate transport to the laboratory within 15 minutes before the contamination. But for most of the specimens, the transport time should not be exceeded to 2 hours. In urine, so in urine is added with preservative boric acid, it is acceptable up to 24 hours. Otherwise, it should be transported within 2 hours. So, urine, when treated with the preservative boric acid, can be used as a sample for up to 24 hours to check for any presence of bacteria. Otherwise, normal urine collected sample must be transported within 2 hours. Next, stool culture. So, the stool specimen should be carried within 1 hour to the laboratory, but we need a transport medium for a stool, that is carry Blair medium. Next, any rectal swabs, if you want to examine bacteria in rectum region, the rectal swabs are used to collect the sample. Up to 24 hours, the transport time is allowed. For anaerobic culture, we want the specimens to be put into Robertson's cooked meat broth or any other anaerobic transfer system and transported immediately to the laboratory. So this medium is important. Robertson's cooked meat broth medium and Caribler medium for stool culture is important. Next, we want to store the specimen before processing. So in the laboratory, so we want wait maximum. We want to wait up to only 24 hours for storage. So if you take blood cultures, it should be incubated at 37 degrees Celsius and any sterile body fluids, any vitreous fluid or suprapubic aspirate from our human body fluids, they must be sterile and they should be immediately plated and incubated at 37 degrees Celsius. Corneal scraping. So the cornea removed should be immediately plated on bedside onto blood agar and chocolate agar. So this is the medium on which cornea is stored. Next one, stool culture. Stool culture can be stored up to 72 hours at 4 degrees Celsius. Urine. Next one, in urine, lower respiratory tract specimen and gastric biopsy. These sample can be stored up to 24 hours at 4 degrees Celsius. So this is you can just go through, and then let us see about direct detection, that is uh, staining techniques, through staining techniques. So first let us see what is heat fixation. So when the flame is gently heated in an air dried film, it is called heat fixation. Methanol fixation, it is used for blood smears. All these are used to detect the presence of bacteria. Okay. Common staining technique includes simple stains such as basic dyes, which include methylene blue or basic fustian, are used as simple stains. So basic dyes such as methylene blue or basic fustian are used as simple stains. So they provide the color difference but impart the same color. So all bacteria will get stained with the same color, which contrasts with the surrounding media. So negative staining. So when a drop of bacteria suspension is mixed with dyes such as Indian ink or nigrosin, so the background will get black, stained black, whereas unstained, bacteria suspension is unstained. So this is useful to demonstrate the presence of bacterial or yeast capsules which do not take up simple stains. That is what negative staining, the name indicates, that the bacteria does not take the stain but the surrounding medium takes the black color stain when Indian ink or nigrosin is used as the dye which is mixed with the given bacteria sample. Next method is impregnation method. So already we have seen simple stain, usage of basic dyes, negative staining using dyes such as Indian dye, Indian ink or nigrosin, that is negative staining. Next, impregnation method, that is in light microscope bacterial cells and structures are too thin to be seen, so they are thickened by treating with silver salts on their surface to make them visible. Only then the bacterial surface is visible on treatment with silver salts to demonstrate presence of bacterial flagella and spirochetes. Next, we are going to see a differential stain. Two stains are used to impart different colors to different bacteria which help in differentiating bacteria. Differential stain, we have seen in WBC last year, first year MBBS, that different types of WBCs will can be identified by using Leishman stain, so which will give characteristic color to the specific WBCs. Here, same here, two stains are used which will impart different colors to different kinds of bacteria and helps in differentiating one category of bacteria from the another. First one is gram stain. So it differentiates bacteria into gram positive and gram negative bacteria. Next we are going to see acid fast stain. So it differentiates bacteria into acid fast and non-acid fast stain. Albert stain. So it differentiates bacteria having metachromatic granules from other bacteria that do not have them. So Albert stain is nothing but bacteria having metachromatic granules or not. Acid fast stain means acid fast or non-acid fast groups. Gram stain is gram positive or gram negative bacteria. So gram staining was developed by Hans Christian Gram in the year 1884. So the gram staining still remains the most widely used test in uh, diagnostic bacteriology. That is important. Although we have many modern diagnostic facilities, gram staining remains the most widely used test in diagnostic bacteriology. First, let us see the basic steps. The first step is fixation. So the smear which is made on the slide from bacteria culture is air dried and then heat fixed. That is important. First it should be air dried and then heat fixed. So we will see the step 1. We first add gentian violet or methyl violet to the given sample and wait for 1 minute. And then the slide is rinsed with water. Crystal violet will stain all the bacteria violet in color. So gentian violet and methyl violet, gentian violet and methyl violet will stain all the bacteria violet. That is important, whether they are gram positive or gram negative. Next step is mordant. Step 2, mordant. We will add grams iodine. Grams iodine is nothing but dilute solution of iodine, which is poured in a slide and waited for 1 minute, and then it is also rinsed. Second time we are rinsing with water. And then next, third step is decolorization. Now we will pour few drops of decolorizer, example acetone. Next, acetone is added wait for 1 to 2 seconds. If methyl alcohol is added, we should wait for 20 to 30 seconds. And then the slide is immediately rinsed with water. Okay, for step 1 and step 2 we will wait for 1 minute, but step 3 we should within seconds we have to rinse with water immediately after adding acetone. So first step, what we saw, we saw that methyl violet or crystal violet should be added. Next step, we need to add gram iodine. Grams iodine is dilute solution of iodine. And next step 3, we will add acetone, which is a decolorizer. So what is the function of decolorizer in this step is it removes the primary stain from gram negative bacteria while the gram positive bacteria. This is very important. Decolorizer will remove the primary stain from gram negative bacteria. Gram negative bacteria do not take up the primary stain, so it will remove. Though it takes the stain, but it can be removed by decolorizer. But gram positive bacteria does not allow the primary stain to be removed, and it will retain the primary stain. That is what the principle behind gram staining. Next, counter stain. So secondary stains such as safranin or dilute carbol fission is added for 30 seconds. It will give pink or red color to gram negative bacteria. So step 4 is not needed. Step 1, step 2, step 3 is enough. Okay. Next we will be seeing about interpretation of gram stain. So gram positive bacteria resist decolorization and retain the primary stain, that is violet. So what is the primary stain is nothing but violet color stain, whereas gram negative bacteria decolorized on addition of acetone and therefore they take counter stain and appear pink in color. Next, let us see the principle of gram staining. So though we may not understand the exact mechanism, but we still have two theories, pH theory and cell wall theory. So cytoplasm of gram positive bacteria is more acidic and hence it can retain the basic crystal violet for longer time. That is the primary stain. Iodine serves as mordant. It combines with the primary stain to form diiodine complex which will retain inside the cell. That is a pH theory. Next one, cell wall theory. In cell wall theory, gram positive cell has the peptidoglycan we have seen earlier, but a peptidoglycan itself is not stained. Instead, it seems to act as permeability barrier. It seems to act as permeability barrier, preventing loss of crystal violet. So it is a permeability barrier. That is the function of peptidoglycan. It prevents the loss of crystal violet, which is present inside the bacterial cell. Okay, so that is what we have seen before. So alcohol is thought to shrink the pores with the peptidoglycan, hence large diiodine complexes are not able to penetrate this tightened peptidoglycan layer in gram positive bacteria. So what do you need? Peptidoglycan, it's a permeability barrier. It will prevent the loss of crystal violet. That is the function. You have to remember that. Gram negative cell wall is more permeable. Thus, it will allow the outflow of crystal violet easily. Okay, because the peptidoglycan layer is thin and it is not tightly cross-linked. We have seen the bridge. I think we have seen in gram-positive bacteria. We have seen one bridge that is called as I think pentaclide bridge. Um, uh, pentaglycine bridge. It makes it peptidoglycan layer very tight. But here in gram-negative cell, it is only through covalent linkage. So it will easily release. It will easily gets decolorized. Next, we will see acid-fast stain. So it is discovered by Paul Ehrlich and modified by Zell and Nelson. So it is called Zell-Nelson technique or hot method. You will see how to do. Smear measuring is prepared in a new, clean, grease-free, scratch-free slide. And the smear should neither be too thick nor too thin. And when it is placed over printed matter, the print should be readable through the smear. Okay, so the smear preparation should be done near a flame. Okay, we will see a steps directly, so we will understand the rest. First, what is heat fixation? So the smear is air dried for 15 to 30 minutes and then heat fixed. First it is dried and then it is heated for 3 to 5 times for 3 to 4 seconds. Coagulation of the proteinaceous material of the sputum will facilitate fixing of the smear. That is what heat fixation. First, let's see step 1, primary stain. So smear is poured with carboyl fistuline 1% for 5 minutes. We have to wait for 5 minutes, and in heating is done by the flaming underneath below the slide until the vapor is seen. So why we are heating the smear? We want increased penetration of the stain into the cell, and we must take care that the smear does not dry out. Okay, so to counteract the drying, more solution of stain is added to the slide is reheated. Okay, in order to overcome the drying, we need to add more solution of stain and the slide is reheated. Next, we want to rinse the slide with tap water until all carbon fission is washed away. Now the smear looks red in color. Next, decolorization by adding sulfuric acid and allowing it to stand for 2 to 4 minutes, and is rinsed with tap water and filtered to drain off the water. So now it appears light pink. A properly decolorized slide appears light pink. If the slide is still red, then sulfuric acid is reapplied again and then rinsed gently with tap water. Next, counter staining. Now this time 0.1% methylene blue is poured onto the slide and you need to leave it for 30 seconds. Then after 30 seconds, just rinse gently with tap water and allow it to dry. That is what counter staining. Then under binocular microscope, we will observe the smear. Okay, so that is what all about acid fast staining. Okay. Okay, so what? So these chemicals are different. First one we had carbon crystal. Then in the next step we added. In the last step we added the sulfuric acid as decolorizer. But in second step, what we added is we added. Okay, there are only two steps. First carbon fission, then sulfuric acid, then methylene blue is added. Second step sulfuric acid, then methylene blue is added. So that is important. So these three chemical reagents are important and might be asked in the exam. Zell-Nelson technique or hot method. Next, how to detect the bacteria? We will be seeing about other methods. So antigen detection. See, latex agglutination test, immunochromatographic test are available which can detect. So these two test names alone, if you remember, it's enough. And then classical example includes detection of capsular antigen. Okay. Urinary antigen detection for pneumococci. Okay, so antigen detection is right now not needed, but you can still remember two tests: latex agglutination test and immunochromatographic test. And you can remember urinary antigen detection and detection of capsular antigen pneumococci, meningococci, and here it is pneumococci and legionella, and direct fluorescent antibody test. You can remember names for detection of Treponema pallidum. So what is the test? This is important. Test for detection of Treponema pallidum. Treponema pallidum is a bacterial which causes syphilis, sexually transmitted disease. So for this, the test used is to detect the presence of Treponema pallidum is direct fluorescent antibody test. Next, let us see molecular diagnosis. See, bacterial DNA or RNA can be directly detected in clinical specimens by PCR. What is the very important concept. Next, culture identification and AST. We will see what is culture media. So what is the culture media? As the name indicates, the microbiological culture medium. It is a medium, either liquid or solid, which contains nutrients to support the growth and survival of microorganisms. Now let us see the constants of culture media. So culture media contains water electrolytes, peptones, agar, then meat extract, yeast extract, and blood and serum. So all these names, if you remember, it is enough. Next, we'll see types of culture media. So first one, based on consistency, culture media grouped into liquid or broth, that means like soup, or semi-solid and solid media. Next, based on the method of growth detection, culture media classified as conventional culture media and automated culture media. So in conventional culture media, they are prepared from nutrients, just aqueous extract of meat, peptone, etc. And the bacteria was detected manually by visual inspection of turbidity or colony morphology. So it is a nutritious medium which contains aqueous extract of meat and peptone. So examples, so the various types, the enriched media, enrichment broth, selective media, differential media, transport media, anaerobic media, and simple or basal media. All these you can identify if you write enough. Next one is automated culture media. They are available for blood and sterile body fluid culture. Okay. So for automated culture media only we have to put blood and sterile body fluid culture, and that growth is directed automatically by the equipment. That is automated culture media, very important. Two media based on consistency, based on method of growth detection, we have seen conventional culture media and automated culture media. Conventional culture media, what it contains is aqueous nutrient sources of meat and peptone. Next, conventional culture media, we will be seeing about in detail about simple or basal media. They usually contain minimum ingredients which will support the growth of non-fastidious bacteria. So what do you mean by non-fastidious? So the bacteria which can grow without any nutritional supplements are called as non-fastidious bacteria. So what are the ingredients present? Peptone water, nutrient broth, nutrient agar, semi-solid medium. Okay, these are all present. Next one we will be seeing about enriched media. Enriched media and basal medium is added with additional nutrients. This blood, serum, or egg. It is called enriched medium. So that's it. So in addition to non-fastidious organisms, they also support the growth of fastidious, nutritionally exacting bacteria, nutritionally existing bacteria. So blood agar. Enriched media includes blood agar, chocolate agar, low flux serum slope, and blood culture media. Blood agar is prepared by adding nothing but 5 to 10% of sheep blood to the molten nutrient agar at 45 degrees Celsius. We want to add 5 to 10% of sheep blood to molten nutrient agar at 45 degrees Celsius. It is a widely used medium in diagnostic bacteriology. Blood agar tests the hemolytic property of bacteria. So partial or alpha hemolysis, green hemolysis, or completed beta hemolysis. So we'll check for these events. So chocolate agar. Chocolate agar is a heated blood agar. Very important. Prepare by adding 5 to 10% of sheep blood to a molten nutrient agar at 70 degrees Celsius. Only the temperature is different comparing to blood agar and chocolate agar. So the RBCs will be automatically lysed and the content of RBCs will be released. So it will change the color from color of the medium to brown color. So it is more nutritious than, there can be difference between blood agar and chocolate agar. So it's more nutritious. The temperature difference also you should know, at which sheep blood is added to the molten nutrient agar, and then it supports certain highly fastidious bacteria. It supports Haemophilus influenzae, that which will not grow on blood agar. This can also be a difference. Haemophilus influenzae is a fastidious bacteria, it needs nutrients. So chocolate agar is more nutritious than blood agar. So Haemophilus influenzae does not grow on blood agar. Instead, it grows on, it multiplies in chocolate agar. Next, low flush serum slope. It contains mainly serum, as the name indicates. So it is used for isolating Corynebacterium diphtheria. Next, blood culture media. They are also enriched media and they isolate microorganisms from blood. That is what blood culture media mean. They may be available as conventional or automated blood culture media. Next, enrichment broth. So these are the liquid media with some inhibitory agents that will allow selective organisms to grow and it will inhibit others. So it will isolate, importance is to isolate pathogens from clinical specimens which also contain normal flora. For example, stool and sputum specimen. Okay, so it will help in isolation of only pathogen from other components. So examples include tetrathionate broth used for Salmonella typhi, which causes typhoid fever, gram-negative broth, which are useful for isolation of Shigella, and two broths are used: gram-negative broth and selenate F broth. Then alkaline peptone water is used for isolating Vibrio cholerae. So examples are important. They might be asked in MCQs. So selective media and all you must learn. Selective media means okay. So I'm going fast. So you have to know all these terms. Selective media, all examples you have to go through. Selective media means it contains inhibitory substances to inhibit the normal flora, and these types you have to remember. The examples, no other way. Selective media. Transport media, they will use the transfer of clinical specimens which are suspected to contain delicate organism, or in delays expected transporting specimen, the bacteria will not multiply in the transport media. It is important. Bacteria does not multiply and they only remain viable. Next, differential media. They differentiate between two groups of bacteria by using an indicator. So McConkie agar is a differential and low salinity medium. It will helps in isolation of enteric gram-negative bacteria. That is the function of McConkie agar. So lactose fermenters, it also differentiates organisms into lactose fermenters that produce pink colored colonies like Escherichia coli, and non-lactose fermenters like produce colorless colonies, example Shigella. That is the function of McConkie agar. So composition is peptone, lactose, agar, neutral red, indicator, and taurocolate. So combination of blood agar and McConkie agar is used by most laboratories for this routine bacterial culture. Okay, next we will go. This is CLED, cysteine lactose electrolyte deficient agar. Okay, so we have to memorize it. Next, anaerobic culture media. So anaerobic media will contain reducing substances which take up oxygen and create lower redox potential. So these media contain reducing substances which will consume oxygen and thus permit the growth of only obligate anaerosis clostridium. So Robertson's cooked meat broth we have seen earlier. It contains chopped meat particles, beef heart. This is chopped meat particles, beef heart, which provide glutathione. Glutathione is sulfidyl group containing reducing substance, and unsaturated fatty acids are also present. Most widely used anaerobic culture medium. This is the significance of Robertson's cooked meat. Okay, so other anaerobic media are mentioned here. Next, we'll go to blood culture media. So with this, we'll complete it so that culture identification. So we'll complete with this. Culture methods are all not needed. Okay. So blood culture media. So removing bacteria from blood is difficult because they are present in lesser quantity in the blood, but on outer environment is present in huge amounts. So to separate bacteria from blood, we use blood culture media. So conventional blood culture media, two types: monophasic medium, it contains brain heart infusion broth, and biphasic medium does liquid phase containing BHI broth and solid agar slope made of BHI agar. So the recovery of organisms in the blood is enhanced by mixing the blood in the broth periodically. If any growth occurs, it can be detected as bacteria or not. So disadvantages are there. So this process is performed less frequently as it is cumbersome. So from monophasic BHA broth, subcultures are made onto blood agar and McConkie agar periodically for 1 week. So there is higher risk of contamination. That is the first disadvantage. While using monophasic BHA broth, opening of the cap of the bottle every time when subcultures are made. So from biphasic broth, so subcultures can be made just by tilting the bottles so that the broth runs over the agar slope, but there is a lower risk of contamination as it obviates the opening of cap. So no need to open the cap of the bottle. Thus they are tilting it out. Okay, from biphasic behavior, this very important. Whether cap is open or not, and contamination potency in monophasic BHA broth and biphasic BHA broth might be asked. Next, automated blood culture techniques. Automated means these are modern culture techniques. So the culture bottles are loaded inside the automated culture system, and the incubated bottles are periodically tilted and automatically every 10 minutes. Every 10 minutes it is tilted so that it allows mixing of blood with broth, which speeds up the recovery. So bottles are periodically monitored for the microbial growth once in every 10 minutes. So how much once in every 10 minutes it is monitored by the instrument. So if it is a positive for microbial growth, then the instrument gives a signal and will give color change in the screen. So composition is tryptic soy broth or brain heart infusion broth, polymeric resin beads which absorb and neutralize antimicrobials present in blood specimen. Then more sensitive, it gives a higher yield of positive culture from clinical specimens, and this rapid, it is a faster method of blood culture media, and it is a less labor intensive as this automatically running machine. So it's more sensitive because it gives a higher yield of positive cultures, and that's it. So examples: back T alert, 3D alert, Virtuo, and back tech. But this is a highly costly instrument, and its inability to observe the colony morphology. Since liquid medium is, we can't observe the colony morphology. This is the disadvantage. That's a very important one. Okay. So remaining methods I'm not discussing because as we don't have time. So as we don't have time, we are going fast. So there is no time to discuss. Okay, we'll go for culture identification directly. Culture methods are also important. Okay, okay, we'll stop with this then. Culture identification. Okay, so that's it. I think remaining all, I don't know whether they will ask or not. There's less chance to be asked in exam, but make sure you're thorough in culture media. Culture methods and culture identification. We'll see if it comes in tomorrow's exam or not. Rest, rest of the topics. Hope you are clear. Since I'm providing lecture only for pathology and pharmacology, I'm going very fast with microbiology since many have requested me to present this video. So we will be going to next chapter in microbiology. Thank you.